Hello and welcome to today's webinar on translating cellular therapies from bench to bedside. My name is Catherine Cook and I will be your moderator today. I work at Nexcelom Bioscience, a company which specialises in the science of cell counting and is now part of PerkinElmer. Now I'd like to hand over to Dr. Anis Kamani, Senior Strategy Leader in Life Science at PerkinElmer, to introduce our speakers.
Thank you, Catherine. Good morning, good afternoon, and good evening, everyone. We welcome you all to PerkinElmer Nexcelom's webinar on cell therapy. Thank you very much for your time to join us today. Combating disorders such as cancer over the years, as you all know, have led to the development of multiple therapeutic approaches. And cell therapy has been one of those promising strategies against cancer. To that end, today we will hear from one of the leading pioneers in the field of cell therapy, Dr. Khalid Shah of Harvard Medical School, who will talk about innovative approaches in developing cellular modalities and its clinical translation. Dr. Khalid Shah is Vice Chair of Research in Neurosurgery at Brigham and Women's Hospital, Harvard Medical School, and also the Director of Stem Cell and Translational Immunotherapy, as well as Principal Faculty at Harvard Stem Cell Institute. Dr. Shah and his group's translational stem cell research has globally been recognized and highlighted, including BBC and CNN. In the second part of this webinar, Dr. Jean Chu, founder and CTO of Nexcelom, will discuss the importance of standards and reference materials and its value in ensuring compliance of cell samples, cell evaluation during cell therapy workflows, and manufacturing processes. Prior to founding Nexcelom, Dr. Chu worked for the 3M company in various roles and was instrumental in invention of the first blue-green semiconductor laser. She has been awarded 34 patents and has to her credit over 58 peer-reviewed publications. Having said that, without further ado, I hand the floor over to Dr. Shah.
Thank you, Anis, for your kind introduction, and I would like to thank PerkinElmer and Nexcelom for inviting me and allowing me to present our work in cell therapies that we've done over last decade or so. We do cell therapies. There are lots of other people, other researchers worldwide who are involved in cellular therapies. There are some bottlenecks in how do we progress cell therapies from bench to bedside. And I'll take you a little bit back into the agitation and the innovation modes. So if you look at the hundreds of social change initiatives over multiple years, the three distinct roles played by those who participate in movements. And the three distinct roles are of the agitator. So unless you are not happy with the status quo, you cannot innovate. So the innovators come into play when they want a change. And that happens in the social change scenario as well, where you sort of are not, you don't agree with what is currently happening and you want to bring a change. And the innovator is born. And then it's crucial for these agitators or the innovators to fully comprehend and respect the models of all stakeholders, the allies, the detractors, and the people who are sitting on the fence. And usually the agitators or who don't agree with the current status are the innovators. So both the roles of an agitator and an innovator are played by the same person. And in many cases, if innovation doesn't go to the next level, it is the orchestration problem, that we have not been able to orchestrate what was innovative. And these are the this is what the focus of the cell-based therapies in current scenarios, that we have so many exciting options for cell-based therapy. There are T cells, there are stem cells, there are cancer cells that you can use for, there are vaccines that we can use. Are we orchestrating this well? And then impact basically requires all three. You need a change, you need agitation, you need innovation, of course, and then you need an orchestration. And we know that innovation without orchestration is more like ideas without impact. So I'll give you a few examples. So if you look at the major cell types, we know that there are embryonic stem cells and there are adult stem cells, and we never thought, that something that is fully differentiated like an adult stem cell would sort of go back and form the basic stem cell that comes from the blastocyst, like an embryonic stem cell. But there has been agitation that was going on since 1976, and that finally gave rise to an iPSC cell when Professor Yamanaka won the Nobel Prize for Physiology in 2012. This was going on since 1976. If you look at the fusion of the adult somatic cell and an embryonic stem cell, and Dolly the sheep came into existence, and the first human iPSCs in 2007. So there was the concept that you can de-differentiate a cell and make it a basic stem cell. So this is one concept, and if you look at the iPSC applications now, this is really wide. You can use this in disease modeling, drug discovery, and toxicity tests. The applications are endless. Similarly, if you look at the CRISPR was adapted from a naturally occurring genome editing system in bacteria. And again, the idea was, can we edit the genes in cells with much more ease? And the history of CRISPR goes back to 1987 till in 2013 we had the successful CRISPR-Cas9 genome editing. And again, the applications are endless. CRISPR is used almost in every modern lab now, and as compared to zinc fingers and talons, sgRNAs that are used in CRISPR system are easier and cheaper to design, and applications, again, you can identify gene functions, you can repair mutations, you can do disease modeling. So these are, again, two concepts, and before I go into the cell-based therapies, where people or researchers were not accepting the status quo. They wanted a change, and it did come. It might have taken 15, 20, 25 years, but it did come. So there was some kind of an agitation somewhere that led to innovation. So now let's go to cell-based targeted therapies. I put them in two different buckets, and you might have heard about this, the autologous and the allogenic cells. Autologous you take from the patient, and these include the T cells, the NK cells, the macrophages, and then the iPSC cells that you can make neural stem cells and the blood cells from this, and you can modify them ex vivo and infuse them back into the patient. From an allogenic perspective, you take healthy individual cells, modify them, and put them into a patient. Though one of the major differences between the two cell types is that allogenic is easily accessible, it can be off the shelf, whereas autologous, the advantage is that it's patient's own, so there is no rejection, there's less rejection. But for the ease of doing therapies and having therapies readily available, allogenic is the way to move forward. Now, from our work, we've used both allogenic and autologous, more allogenic where we used engineered stem cells from a healthy individual and modified them and tested them in our mouse models. And then the iPSC-derived neural stem cells, and now we're making NK cells. So we have published extensively on iPSC-derived neural stem cells. And then from the autologous side, we pioneered the use of engineered cancer cells. And then we have some compelling data on the CAR T cells and the combination of engineered stem cells and CAR T cells. So our focus has been cancer. Most of the times we focus on the primary and the metastatic tumors in the brain. And if you look at the two images, the one on the left is one of my last author papers. I think it came out in 2010 PNAS paper, where we looked at the migration of stem cells to the tumors in the brain. The green are the tumors, red are the stem cells. And then almost 8-9 years later, we sort of pioneered the use of cancer cells to treat their own types. And I think what happened in those 8 years was that we envisioned that technology will develop in parallel. I mean, we would not have been able to do the studies with engineered cancer cells if CRISPR hadn't evolved, if lentiviral vectors hadn't become better and easier to use. So technology is evolving, and as an innovator, you have to think that technology can be applicable to any idea that you have, and you have to count that in to orchestrate it better. That's another example, but I'll go into the details of both of these sort of bigger ideas that we had using stem cells to target tumors and using cancer cells itself to target their own tumors. So bigger focus is that we take tumor cells and tumor-associated endothelial cells and immune cells, and we look at receptors that are specifically expressed on these tumor cells and the cells in the tumor microenvironment. And then we engineer stem cells with therapeutics or ligands or antagonists or agonists that can bind to these receptors on the surface of tumor cells or immune cells or endothelial cells. So we have not only made stem cells releasing proteins, but also oncolytic viruses, which I'm not going to go into the details in the interest of time, and immune modulators, again, very less. I'll talk a bit less about that, and some engineered CAR T cells, but we have made cancer cells that I'll talk a little bit more about. So this is one of the examples where we use stem cells to release a ligand which is binding to the receptors on the tumor cells, which are in red. And what you will see over time is that you won't see the ligand, but you would see the killing of the red cells over time. This is a 54-hour movie, and you will see that what you're seeing is that the green cells remain unaffected. So the ligand is being released from the green cells. It's binding to the receptors on the red cells and killing the red cells. These are the tumor cells. Now, we've published extensively on this, and others worldwide have published on stem cells releasing therapeutics and killing tumor cells. The question is, again, the orchestration part. How do you actually translate this into clinics? And one of the tumors that we have been extensively looking at is GBMs, glioblastomas in the brain, patients with highly malignant glioblastomas. 80 to 85% of the patients get tumor resection. This is the first-line therapy followed by chemo and radiation. And what you see here is a full-fledged tumor and a resected tumor. The issue is that when people do get glioblastomas, unfortunately, they need to be resected. Once they get detected, the operation has to be done within the first week. So we don't have much time to prepare patients' own cells and treat them with their own cells. So we've done phantom studies with patients who have had GBMs and took their MRI images pre and post resection. And all we wanted to do is to look at this cavity. What is the cavity size? Because we know that drugs don't cross blood-brain barrier efficiently. Stem cells don't cross blood-brain barrier efficiently. So we wanted to go locally. So our interest was, can we actually treat these remaining tumor cells in the cavity? And we've done, again, quite a bit of work. And so we did calculate cavity sizes and build a mouse model of resection that mimics these patient-derived models. So what we did is we took these stem cells and encapsulated them in the biocompatible hyaluronic acid-based gels. And what you see is the green stem cells, again, releasing a ligand that binds to the tumor cells, and also cells migrating out of the gel and sort of reaching far away from where the actual resection site is in the brain, and then treating and killing all the red cells. Now, this works very well in culture, and we've shown that in vivo, when you take these green cells that are releasing the ligand to kill the red cells, if you don't encapsulate them in the gel, they don't survive long. But if you encapsulate them in these biocompatible gels, they survive long. And if they are releasing the ligand that's binding to receptors on the tumor cells, you actually get 100% survival in mice. Yeah, so we also created bimodal stem cells that are co-expressing a safety kill switch, HSV-TK and TRAIL, and shown that we really wanted to make it safe if we would take this into patients. So a stem cell that is releasing the ligand TRAIL now also has a kill switch, HSV-TK, whose expression selectively sensitizes cells to ganciclovir, and it can also be used as a PET imaging agent. So we can now kill the stem cells when we want to kill them after they have treated the tumor cells, and we can image this by PET imaging as well. Now, the other sort of aspect that we included in this study is, can we detect, because we know that TRAIL binds to death receptor 4 and 5, and some cells are resistant to TRAIL. One of the reasons is that the receptors for death receptor 4 and 5 are not on the cell surface. So we created a system with Dan Haber and Mema Toner at MGH, where we could actually look at circulating tumor cells in the blood of patients having brain tumor, and also the mice that were implanted with these patient-derived cells, and using this team cocktail. So we could actually look at circulating tumor cells, but now we've taken this to the next level, which is now the paper is accepted in Nature.com, where we can detect receptors on these circulating tumor cells. So not only in mice bearing these patient-derived tumors, but also in patients. If you take a patient who has been detected with GBM, if you take the blood of these patients, we can actually look at we can isolate circulating tumor cells, and we can look at death receptor 4 and 5 in these tumor cells. So the bigger idea is that so let's assume a patient gets detected with GBM, has to be operated within the first week. We'll have blood within the 24 hours of detection. We isolate CTCs and look at the receptors. We'll have our cells, allogenic MSCs banked. We already have the GMP grade ones, and we can, so between this time, a week, let's say a week, we can actually take these stem cells, thaw them, and have them ready for the implantation at the time of surgery. So we'll encapsulate them in the gel, and we'll place them at the time of the tumor surgery within the cavity that has been created by the surgery. We can take this, so we've spoken to FDA about this a couple of times. We are about to submit an IND for this, and hopefully we'll have a clinical trial soon. The question arises, what happens to the cells that are resistant to TRAIL? Can we sort of sensitize these cells to TRAIL-mediated apoptosis? And we've done a number of studies where we've used temozolomide to upregulate TRAIL receptors and sensitize them to TRAIL. We've used PI3 kinase inhibitors, but we wanted to build a molecule that has the sensitizer and the killer in the same cell. And so we looked at EGFR nanobodies. These are the smallest functional fragments of a naturally occurring single chain antibody, and we cloned these in collaboration with Paul Hanegel at the University of Utrecht. we made stem cells releasing bivalent nanobodies and show that they can downregulate EGFR, they can downregulate phospho-AKT, and when we fused the TRAIL molecule to the C terminus of this bivalent nanobody, luckily for us, the EGFR and DR4, DR5 receptors are in the close proximity. That's what our FRED studies show. And then when this molecule binds to both the receptors, the DR4, 5, and EGFR, we see apoptosis happening in semi-resistant TRAIL cells. And if you use cetuximab that blocks EGFR, then there is no apoptosis. That implies that both nanobody and TRAIL is important. And if you knock out DR4 or DR5, then you also don't see any apoptosis. We've done quite a bit of studies with GBM models, but we've also done a lot of studies in brain metastases models. And this is a breast to brain metastases model where you don't get solid nodular tumors, single deposits. When cells go from breast to brain, they have multiple deposits in the brain, like you see here in the MRI and the cartoon as well. So we've sort of looked at all these metastatic tumors, the basal-like, the HER2, the luminal A, luminal B breast cancers, and looked at EGFR and DR4, 5 expression. And we see that EGFR is highly expressed and DR4, 5 as well. But when you look at the match pair between the primary tumor as well as the breast metastatic tumor in the brain, you see huge upregulation of EGFR and also DR5 in these cells, in these tissues. And we've taken all these 10, 15 lines and looked at EGFR, DR4, DR5, and we see, again, upregulation of EGFR, DR4, DR5, both at the cell surface level by flow and by Western blotting as well. So this is a paper published in Science Advances last year. I don't want to go into the details of all the in vivo, in vitro experiments, but in vivo we show that if you resect some of these breast tumors in the brain, metastatic breast tumors, which in very small cases happens, we can actually increase survival in mice. And then if you look at this very hard model, we built a number of mouse models, leptomeningeal metastases model, where you inject cells intrathecally, and you see that the green stem cells are tracking the red tumor cells and again increasing survival. We've taken this now to the next level where we're looking at immunotherapies with stem cells releasing immune modulators and killing agents like TRAIL and nanobody TRAIL. I want to shift gears to two little stories that we have. One, if you look at the trends in global immuno-oncology, cell therapies are really moving at a very, very rapid pace. And so we sort of had to shift gears because most of our studies was done in immune competent or immune compromised mice. We had to build mouse models that were from mouse tumors. So if you look at the GBMs, GBMs are one of the coldest, immunologically cold tumors. The reason is that they have highest number of myeloid-derived suppressor cells, and they have low degree of T-cell infiltration among all the different 6 types that were identified from 33 cancer types and 10,000 TCGA tumors. And so we built models where we've looked at, the mouse tumors, mouse GBMs in syngeneic mice or immune competent mice. And we've identified immune active and immune inactive tumors, immune suppressive tumors. And if you take the immune suppressive tumor and resect the tumor, you see huge infiltration of T cells, great reduction of myeloid-derived suppressor cells that are in the core of the tumor, and again, infiltration of NK cells and the CD4 and CD8 T cells. So we, again, just an example, we are looking at immune modulators and cell killing agents, and one of the molecules that we looked at is interferon beta, that induces apoptosis as well as acts as an immune modulator, and putting our stem cells in the resection cavity. And what we show is that there's huge difference in survival in these immune competent models as well. And we're taking this to the next level where we're now using immune checkpoint inhibitors, which are scFv fragments of either PD-1 or PD-L1. And this is an ongoing work which should come out later this year or early next year. The last part where I want to spend 2-3 minutes is our cancer cell killing cancer cell story, which we published a few years ago. The idea was that we knew that cancer cells love to home to their own tumor types, and they love to go elsewhere, but because the vasculature is not leaky, they don't end up anywhere else. Early on, they end up mostly in their own tumors. And we thought, why not to modify these cancer cells, because they were homing to their own tumor types, and use them to kill the tumors, their own tumors. And one of the advantages was that there's patient specificity, and they're also natural sources of tumor-associated antigens. And the other advantage was that they proliferate well, so the ease of gene editing and gene engineering and ex vivo expansion is much, much easier. So long story short, so what we did is we took a therapy-sensitive tumor cell. We knocked out its receptors, for example, TRAIL sensitive tumor cells. We knocked out DR4, DR5 to make them therapy resistant. And then we engineered them to express the ligand, or TRAIL in this case, and a safety kill switch. And then TRAIL would kill the original therapy-sensitive tumor cells, and we will ultimately kill the therapy-resistant tumor cells by activating the kill switch. So here are some of the data where if you knock out DR4, you get 50% resistant cells. If you knock out both DR4 and DR5 in tumor cells, you get almost 100% resistant tumor cells. And we've done this not only in brain tumors, but also breast cancers, prostate cancer, and a number of other cancer types. Now, if you take the green are our therapeutic cancer cells, they're releasing the therapeutics. And if you look at a few green cells can kill a number of red cells, not only in nodular tumors, but also in invasive tumors. And then the idea is, can we kill these green cells by activating the kill switch? And here we show this in the gel, where gel encapsulated therapeutic tumor cells are actually killing the red original tumor cells, and we can activate the kill switch both in vitro and in vivo, and we can kill these green cells ultimately. And we've shown this again in a number of models. One of the models is the brain tumor resection model, where you resect the tumor and implant the therapeutic cancer cells now in the biocompatible gels, and show killing not only of the original tumor cells, but also the therapeutic cancer cells when we activate the kill switch. We've taken this again to the next level, because this was all done in immune compromised mice, and unfortunately in immune competent mouse, the tumor cells, the mouse tumor cells, are not sensitive to TRAIL, so we had to switch to an immune modulator. So we actually have two immune modulators that we knocked out the receptors, as I showed before. Now we have two immune modulators released from the therapy-resistant tumor cells and the kill switch. The idea is to modulate immune response locally, activate programmed cell death, and then also activate the kill switches that would induce bystander effect. And then you will have tumor antigens exposed, and you will get immunological memory. So not only killing of tumor cells, but also immune modulation that might have some kind of a vaccination effect. And the data are there, and so hopefully, again, in a year or so, we should be able to reveal those data, and hopefully by then it will be sent to publication as well. So these are sort of different concepts. Again, I want to end with the theme of sort of innovation, and then agitation, innovation, and orchestration. And here is the orchestration part. How do you actually translate therapies from bench to bedside? And again, patient becomes the key aspect of this. The molecular profiling of the tumors, and the access to the tumor cells, the access to GMP facilities ultimately after you have designed therapies. And for us, I think clinically relevant mouse models are a big part of orchestration. And if you don't have those well-oiled, sort of clinically relevant models that you can test your therapies on, it's hard to actually translate therapies into the clinic. So I want to leave with the acknowledgements that this work would not have been possible without my team that is pretty international. We take pride in having at least 10, 12 nationalities in the lab at one given time, and also our collaborators within Boston, outside Boston, and our funding agencies. Thank you so much.
All right. Today, I'd like to share with you the other element of translation, which is be able to make the cells to the regulator's requirement so we can safely treat patients. We have heard the very exciting scientific advancement in the cell gene therapy as shared with us from our last speaker. All this excitement, in order for us to translate that into clinics to serve patients, we have to overcome some challenges. And a lot of times, these exciting new therapies involve scientific and manufacturing novelties. Those are the things never been done before. We also heard Dr. Shah talking about lack of preclinical testing models, as well as regulators have new expectations. So it's in the new frontier. Fundamentally speaking, the challenge stems from cell therapies are living drugs. They are complex, heterogeneous, and dynamic. So for Nexcelom, we have been focusing our effort on being the innovator and use our expertise in the science of cell counting. We've been doing that for many years, and indicated by over 14 cell counting systems used in thousands of labs. We serve single sample workflow as well as multi-sample high throughput automated workflows. So what do we mean by standardized cell counting measurements? They are actually very important for cell gene therapies according to FDA. It is used as part of the characterization of safety, efficacy, purity, potency, identity, and the quality of cells. It is used both for product and process development. And as part of quality control test panels for source materials, intermediates, and the products. It is important to use cell count and viability as a part of reproducibility between lots, as well as work on comparability after manufacturing changes. So according to FDA, cell counting is crucial during all stages of cell-based product development. Before you can file for R&D, which is a permit to start running clinical trials, a lot of manufacturing quality-related information has to be submitted before they give you approval. After that, when you are doing R&D, phase 1, phase 2, phase 3, until they give you market approval, there's a lot of work goes into analytical procedure development. So here is the example of process. Preclinical cell counting method need to be selected, developed, optimized. In phase 1, there is a method qualification and start to set some release criteria. During phase 2, the assay optimization happens and validation acceptance criteria are considered, parameters are proposed. During phase 3, validation has to happen and before they can approve any drug for marketing. So accuracy and precision are two key parameters for cell counting method selection. By definition, accuracy is closeness of agreement between a measured quantity value and the true quantity value of a measurement. The precision comes with closeness of agreement between measured quantity obtained by replication. Illustrated in these targets, if we can always get to the center of it and with very tightly hitting the target, means we have accuracy and the precision. If we can hit the target but scatter around it, we have accuracy but a little less of precision. If we are closely clustered outside the target, we are precise but we're not accurate. If we are scattered all over the place, that means we don't have either. Accuracy is ideally evaluated using a reference method or reference material which has a true value. And the reference material by definition is material sufficiently homogeneous and stable with a reference value. So now we have a dilemma because live cells are dynamic and they cannot serve as reference material. So this is the background where the FDA is actively engaged with ISO standard development. So far, there are 2 published counting standards. Part 1 is general guidance on cell counting method. Part 2 is experimental design and the statistical analysis to quantify counting method performance. I would recommend anybody work on CMC for cell therapy and purchase the standards, and they give very good guidance. So in Nexcelom, other than participating in the standard development in the last 5 years, we thought a lot about how to practice it. So today, during the time limitation, I will not be able to go in depth. Please contact us. We have a lot of in-depth material on how to do it. Here, I'm just going through a few key success factors. We already described briefly, research use cell counting is different than cell counting procedures submitted to regulatory agency for drug approval. FDA care about quality in cell counting for cell and gene therapy. The cell counting method selection needs to fit for purpose, not one size fit all. So this is a slide borrowed from Dr. Gibson from NIST. She was talking about when you do fit for purpose cell counting method selection, you actually think about you're trying to count some kind of cells, AA, in a matrix of BB, which you support the cell with, and using a particular counting method, CC, for DD as the purpose. Are you doing release testing for dosage evaluation? Then that's very, very critical measurement, or it's in process control. Fit for purpose has a lot of worksheets and the process to go through. If you're interested, contact us. So the next key success factor involves we need to understand our cell samples. We identify best cell counting methods before we select the cell counting system. Even the cell counting system is going to include the reagents, consumables, counting devices, and the software. So in this picture, I show you by using fluorescent nuclear staining, we can identify and count nucleated cells in apheresis samples without lysing red blood cells. These green color are nucleated live leukocytes. The orange color indicate dead ones. Then you're going to notice there's a lot of other particles, and these are either red blood cells or platelets. If you're doing bright field trypan blue measurement, either using manual counting or by cell, you're going to overcount the live cell for the sample. So this is another indicator image of trypan blue stained mouse lymphocytes. So you can look at the live cells are these shining spots, but the blue spots are very diffuse. Some of them are diffuse and very large. And I will show you a video of trypan blue damaging these dead or dying immune cells. So in this example, we aged some Jacquet cells, and then we pipette the cell into an imaging slide. The next step, we put some trypan on the inlet port, allow it to diffuse and interact with Jacquet cells, and we film it in action. So if you look at the zoomed area, when the trypan diffuse in, see what happens to this particular cell. It gets expanded by interacting with trypan. This is a more magnified case. You're actually seeing the explosion of the cells. It's going to repeat. So what happens with this situation? And this solves the mystery of sometimes we have unrealistically higher viability when using trypan blue cell counting method. So next one, I'm going to switch to telling you about the quality of cell counting results are determined by the entire cell counting process, not just the cell counter itself. We have seen too many people focus on selecting the cell counter. However, how do you collect sample from your original vessels? How do you mix and prepare them? And how do you do data analysis after you have the cell images? They all contribute to the counting result and contribute to the quality. So the next step is about design experiments to obtain multiple quality parameters for cell counting method evaluation and development. ISO cell counting standard part 2 has a lot of details in that, and we have a lot of worksheets and protocols related to it. After you get the cell counting data, it is important to use the uncertainty approach to experimentation and the statistical method to understand the result. What kind of variability do we expect? And also to set acceptance criteria. This is another area statistical knowledge is important. Since we talked about lack of accuracy standards for live cells, it is important to do orthogonal experimentation to help us understand which selected cell counting method are more accurate. So the next one is more of a practical matter. Most of us have not done a lot of cell counting measurements on different kinds of samples and with different experimentation, except Nexcelom scientists and field application and engineers have done hundreds of these measurements. We have a bank of data can share with users. Just let us know what you need. So it is important to know, even we think cell counting is the most fundamental and straightforward measurement, it's actually not. So to do it with high quality and reaching out to us, we'll share with you on the knowledge. So we continue to work on viability in collaboration with NIST, and it is another critical quality attribute for cell therapy. So I can't emphasize enough, let us help. We have done worksheets, webinars, different tools to use for increased cell counting quality. Before I finish, just want to mention not only these systems are used in GMP-related environment, it started from basic research and all the way through the process. If you want to translate cell therapy fast, start identify good measurement tools and use it throughout the process, save a lot of time and resource. Thank you.
Thanks, Dr. Shah and Dr. Chu for your presentations. Thank you very much. We've reached the end of our scheduled time now, so I would just like to thank the audience for joining today's webinar. And I'd also, of course, like to thank our speakers for their presentations today. I'm sure everyone found it useful and interesting. Thank you very much.